This lecture is about important molecules in molecular biology. Now I'm only going to talk about a few key molecules because they're the ones that we talk about throughout the course, and it's important that you know their names and you know what they are. There are thousands of molecules that we could talk about. They're all important, so I don't mean to imply by saying important molecules in molecular biology that these are the only important ones, but they're among the most critically important molecules in determining how your genome functions. And if you get confused or you worry whether or you wonder whether this is that important, just remember that every one of these molecules is in every cell in your body. If you had a powerful enough microscope, you could you could just point it at your skin right now and zoom in and you could see all these molecules I'm going to talk about. So DNA is the molecule that comprises all of our genetic material, and it's comprised of 4 different nucleotides. A, G, C, and T is how we write them. They're actually named adenine, guanine, cytosine, and thymine. And biochemically, they have slightly different structures. Adenine and guanine are called purines, and they have a 2-ring structure shown here. And thymine and cytosine are pyrimidines, and they have a 1-ring structure. And you don't need to know this structure or remember this, but it's good to know that the C and T are kind of similar to one another and a little smaller, and the A and G are kind of similar to one another and a little bit bigger. So the way the DNA is constructed is that these molecules bind together in a very specific way. A's always bind to T's, and G's always bind to C's. And this is true across all living things. Everybody has the same DNA, and we all have the same structured DNA. So this rule is very useful because it means, and this was discovered way back in the 1950s when Watson and Crick discovered the structure of DNA, they immediately realized that this binding property means that when you have one of the strands, you also know what the other strand is. So if I give you one of the strands of DNA with A's, C's, G's, and T's, you know what the other strand is because everywhere there's an A on one strand, there must be a T on the other strand, and correspondingly everywhere there's a G on one strand, there must be a C on the other strand. So this provides a mechanism, as Watson and Crick famously observed when they discovered the structure, for how DNA copies itself and passes itself on from one generation to another. So the structure of DNA then in these long strings gets put together in this famous double helix structure. So the molecules, the A's bind to T's and the G's bind to C's, and you build these long ladders, and the ladders are twisted in a long helix. And every one of your cells has all of your DNA in it in these structures, and as we've said before, the DNA in our genome is organized into 23 chromosome pairs. Each of these chromosomes is a very, very long string like this. The longest chromosomes in the human genome are on the order of 250 million nucleotides long. So they're extremely long molecules, very tightly coiled up and packed together inside of the nucleus of every cell in your body. So the way that we're going to write the data, the DNA sequence itself, looks like this. So we'll write A's, C's, G's, and T's. We won't write these chemical structures. We abbreviate it with these 4 letters. And DNA actually has a direction, a strandedness, and we call this based on the biochemical properties. One end of the DNA is the 5' end, and the other is the 3' end, and that has to do with the structures of those biochemical molecules. But you don't really need to remember that. Just remember we always write it in the same direction, where 5' is always first and 3' is always second, and we generally write the opposite strand the other way around. So the strand that goes 5' to 3', because we're writing things that way, we call that the positive or the plus strand. And the other strand, the reverse complement, is the negative strand. So another critical molecule for how our body works and how our genomes work is RNA. So RNA is almost exactly like DNA, except for a couple of important differences. The most obvious difference is that we don't have the T anymore. We don't have thymine. Instead, we have uracil. So when DNA gets copied or transcribed into RNA, the A's get replaced by A's, G's get replaced by G's, C's get replaced by C's, but T's get replaced by U's, or uracil. And then you build an RNA molecule which, unlike DNA, is single-stranded. So RNA molecules are not double-stranded, although they can form double-stranded complexes. But in general, RNA is single-stranded, and it's from this RNA template that we create proteins, which are the other critical molecule in the cell. So RNA has, again, the same similar biochemical structures. The uracil, or the U molecule, is very similar to the T molecule. So we write RNA the same way we write DNA, 5' to 3', only we're going to replace all the T's with U's. So if you see a string of letters and there are U's in it instead of T's, you know immediately that that's RNA, not DNA. So an important distinction genetically is that DNA is the stuff of inheritance. DNA is what the cells carry with them from one generation to another. Whenever a cell divides, it creates DNA that replicates the DNA in the original cell. The RNA is used as a template to make proteins, but the RNA is not actually the stuff of inheritance. However, it is in most cases an identical copy of the DNA that it was formed from. So we use these molecules to encode how the cell works. So DNA is basically a program that we read out, and the readout program starts with RNA, and then it goes to make, and the RNA is then used to make proteins. So proteins are also long molecules, not nearly as long as DNA. They're typically hundreds or sometimes thousands of amino acids long. So amino acids are more complicated molecules that, there's a picture of one here, that are strung together as well to make proteins. And the translation rule, the translation rules were worked out back in the 1960s in some groundbreaking molecular biology work, really at the very founding of the field of molecular biology. Scientists figured out essentially one codon at a time how you translate RNA into proteins. So I just use the word codon. So the way that RNA gets turned into proteins is that every combination of 3 of the letters of RNA encodes an amino acid. So there are 64 possible such combinations. Of those 64 combinations, 61 of them encode amino acids, and 3 of them are stop codons. So the translation machinery reads along the RNA molecule 3 nucleotides at a time, and for every 3 nucleotides, it creates an amino acid, and those are built together into a long string of amino acids, which is what we call a protein. And when the RNA translation machinery hits one of these stop codons, one of these 3 special codons that don't encode anything, it stops, and that's the end of the protein. So that's how it knows. So we write protein sequences also in a particular direction. And here's just showing a translation of a particular set of 9 nucleotides producing 3 amino acids. And we write proteins in the 20-letter alphabet we use to abbreviate the amino acids. There are 20 amino acids that comprise essentially all of our proteins. So actually, to tell the whole story, there are more than 20 amino acids. There's actually 22. The 21st amino acid was discovered not that long ago, and the 22nd amino acid also not that long ago. And these amino acids are primarily used in other forms of life besides humans. So there are a few exceptions to almost every rule in biology, but in general, the way to think about human biology is that we have 64 possible codons, 61 of them encode amino acids, and they encode exactly 20 amino acids. That 21st amino acid that was discovered turned out to be one of the stop codons that's once in a while used to encode an amino acid.